Good evening, and welcome to Dreamy Academy. Tonight we begin a quiet journey into the very fabric of existence, exploring the profound questions that biology still holds close, like secrets whispered in the oldest forests. As we begin, I'd love to know where you're watching from, your city, your country, perhaps even what the night sky looks like outside your window. If you enjoy these peaceful explorations of science, please take a moment to like the video and subscribe. It truly helps this little corner of quiet curiosity grow. To those already subscribed, thank you sincerely for being part of this slow and thoughtful adventure. And if you'd like to be gently reminded when new chapters unfold, the notification bell is there, waiting. Every breath you take, every beat of your heart, every thought that ripples through your mind, these are all echoes of a story far older than humanity, older than mountains, older even than the sun that warms our planet. It is the story of life itself, a dance of chemistry and energy that began in the deep past and continues in every cell of your being. We will journey into the unsolved questions of biology, not as problems to be conquered, but as invitations to wonder. For in these lingering mysteries, these elegant gaps in our understanding, we find the deepest beauty, the most profound elegance of the natural world. These are not puzzles to rush through, but landscapes to explore with the quiet patience of twilight, allowing understanding to unfurl like a delicate blossom. To contemplate these questions is to feel the vastness of time and the intricate precision of existence, a gentle overwhelming of the mind that settles into peaceful awe. The world of biology is a tapestry woven from countless threads of discovery, yet there are patterns within it that remain just beyond our grasp. Threads that disappear into the mists of deep time or into the quantum hum of the very small. It is in these places, at the edges of our knowledge, that the greatest wonder resides. We will allow our minds to become pleasantly saturated by the interwoven tapestry of understanding, lulled by the rhythm of discovery itself, as we drift through the museum of life's natural wonders at twilight. Have you ever paused to truly consider what makes a living thing, well, alive? A stone simply is, a quiet arrangement of minerals enduring the passage of time without complaint or desire. A river flows, its currents carving valleys, shaping the land with relentless purpose. Yet it does not live. It is a process, a beautiful, ceaseless movement of water, but it holds no internal flame. Yet a tiny bacterium, invisible without a microscope, actively seeks nutrients, grows, divides, and responds to its world with a subtle, intricate intelligence. What is that fundamental difference? What is the spark that separates the animate from the inanimate, the inert crystal from the simplest cell? This question, as ancient as philosophy itself, remains one of biology's most profound and beautiful mysteries, the very origin of life on Earth. Let us begin by considering what we often take for granted as the hallmarks of life. A living organism, we might say, metabolizes. It takes in energy and matter from its environment, transforming them to sustain itself. It reproduces, creating copies or variations of itself. It grows and develops, following an internal blueprint. It responds to stimuli, sensing and reacting to changes in its surroundings. It maintains an internal balance, a steady state known as homeostasis, even as the external world shifts. And crucially, over generations it evolves, adapting to its environment through the patient, relentless pressure of natural selection. These are the familiar characteristics, the well-worn paths of definition. But the boundaries, like the edges of a cloud at dusk, can blur. A virus, for instance, carries genetic information and evolves, yet it cannot metabolize or reproduce without hijacking a host cell, a mere whisper of life. Prions, even simpler, are just misfolded proteins that can induce other proteins to misfold, a kind of self-replication without any genetic material at all. And a crystal, growing in a saturated solution, exhibits order, a kind of self-assembly, yet we do not call it alive. Life, then, is not merely a collection of molecules, but an emergent property of a highly organized, self-sustaining chemical system, one that has learned to remember itself and to change over time. It is a dance of chemistry, a self-sustaining cascade of reactions, fueled by energy and built from common elements. The stage for this dance was set billions of years ago, on a young Earth far different from the blue-green world we know. Picture, if you will, a tempestuous landscape, cloaked in an atmosphere rich in methane, ammonia, water vapour and carbon dioxide, but almost entirely devoid of free oxygen. Volcanoes spewed forth molten rock and gases, lightning split the dark skies, and fierce ultraviolet radiation from a young sun, unimpeded by an ozone layer, bathed the planet. The oceans were warm, perhaps even hot. A vast chemical laboratory where simple molecules could mingle and react. This was the primordial soup, a term coined to describe the conditions under which life's first stirrings might have occurred. Here, in this ancient cauldron, the basic building blocks of life, amino acids, nucleotides, simple sugars, could have formed spontaneously. The famous Miller-Urey experiment in the 1950s showed that by simulating these early Earth conditions in a flask, with a mixture of gases, water and electrical sparks, amino acids could indeed be generated. This was a profound revelation, suggesting that the very components of life were not mystical, but could arise from the patient laws of chemistry. Beyond the surface waters, another compelling scenario unfolds, deep beneath the waves, at hydrothermal vents. These fissures in the ocean floor spew forth superheated, mineral-rich water, creating unique chemical gradients and protected environments, perhaps ideal for nurturing delicate reactions away from the harsh surface radiation. The true enigma, however, lies not just in forming these building blocks, but in arranging them into complex self-replicating systems. How did individual amino acids link into proteins? How did nucleotides assemble into long chains of genetic information? And which came first? The genetic information, like DNA, or the catalytic machinery, like proteins, that reads and enacts that information? This is the classic chicken and egg problem of abiogenesis. DNA, the blueprint of life, requires proteins to replicate itself and to build cellular structures. Proteins, the workhorses of the cell, are encoded by DNA. Here, the RNA world hypothesis offers a beautiful and elegant solution. Ribonucleic acid, or RNA, is a molecule closely related to DNA. But RNA possesses a remarkable dual nature. It can store genetic information, much like DNA, and it can also catalyze chemical reactions, much like proteins. Imagine a molecule that could both carry the recipe and bake the cake. In a primordial world, RNA could have been the central player, evolving to replicate itself and to perform rudimentary cellular functions, eventually giving rise to both DNAs and proteins. The beauty of this idea lies in its simplicity. A single type of molecule could have bridged the gap between inert chemistry and the first stirrings of biological complexity. Yet, even with RNA, a critical step remained: compartmentalization. Life, as we know it, is not just a collection of molecules. It is a bounded collection, separated from its environment by a membrane. This membrane allows for the creation of an internal environment, distinct from the outside, where specific chemical reactions can be concentrated and regulated. Lipids, the fatty molecules that form cell membranes today, have a fascinating property. When placed in water, they spontaneously self-assemble into spherical structures called vesicles or micelles. These tiny bubbles, or protocells, could have encapsulated random assortments of early molecules, including RNA. Within these enclosed spaces, the chances of beneficial chemical reactions occurring, and even for rudimentary selection to begin, would have dramatically increased. A protocell that happened to enclose an RNA molecule capable of replicating itself, and perhaps catalyzing the synthesis of more membrane lipids, would have had a distinct advantage. The implications of this journey into life's origins are vast and humbling. If life can emerge from fundamental chemistry under specific conditions, if the universe has an inherent tendency to arrange matter into self-sustaining, self-replicating patterns, then the possibility of life beyond Earth becomes not just a poetic dream, but a reasonable scientific inference. The same cosmic chemistry that gave rise to us might be playing out on countless worlds across the galaxy. This grand narrative of abiogenesis, though still incomplete, reveals the universe as a place of profound creativity, where molecules can, given enough time and the right circumstances, learn to remember themselves, to grow, and to evolve. It is a testament to the power of emergent complexity, a reminder that the most intricate wonders can arise from the simplest ingredients. There is a particular comfort in this understanding, a sense of belonging to a cosmic process that is both ancient and ongoing. So we've touched upon the very first stirrings, the potential moment when chemistry learned to dance, when the inanimate whispered its first living breath. We've considered how simple molecules might have organized themselves into self-replicating systems, eventually encapsulated within the delicate boundaries of protocells. But once that first spark ignited, once a simple self-replicating entity came into being, how did it lead to the incredible, breathtaking diversity we see today? How did a single, simple ancestor branch out into every creature, every plant, every fungus, every microbe that has ever graced this planet? How does the subtle pressure of the environment sculpt forms and functions with such exquisite precision? That question leads us into the patient, powerful story of evolution, a tale of adaptation and change across unimaginable timescales. Having journeyed together through the primordial mists where the very first sparks of life ignited from the raw elements of a nascent earth, we now stand at the precipice of an even grander mystery. How that singular, nascent flame diversified into the incandescent tapestry we call the biosphere. From that humble, self-replicating entity, a last universal common ancestor, or LUCA, emerged a lineage destined to unfurl into the breathtaking kaleidoscope of forms that have graced our planet for billions of years. But how precisely did this happen? What were the pivotal moments, the profound transformations that allowed life to ascend from mere molecular machines to the astounding complexity of an oak tree, a whale, or indeed us. As we drift deeper into the dreamy academy's quiet halls, the question that now beckons us is not merely that life diversified, but how it orchestrated such magnificent complexity from such simple beginnings, and what profound enigmas still shroud these evolutionary leaps. We observe the vast, intricate tree of life, its branches reaching into every conceivable niche, yet the roots and early trunk remain shrouded in a fascinating twilight, inviting our gaze and challenging our understanding. Imagine, if you will, the early Earth after Luca. For perhaps 1 billion years or more, life remained predominantly a microbial affair, a hidden world of single-celled organisms, masters of biochemical ingenuity, yet outwardly simple. These prokaryotes, bacteria and archaea, thrived in diverse environments, from scorching hydrothermal vents to frigid polar seas, silently shaping the planet's atmosphere and geology. They perfected the art of survival, developing myriad metabolic pathways that still underpin all life today. But then, a truly revolutionary event transpired, a merger so profound it would forever alter the trajectory of life, the genesis of the eukaryotic cell. This transition from the simple, anucleated prokaryote to the complex membrane-bound eukaryotic cell with its specialized organelles represents one of the most significant evolutionary leaps, a quantum jump in biological organization. How did this intricate internal world, complete with the nucleus housing its genetic treasures, mitochondria powering its every function, and in plants, chloroplasts capturing the sun's energy, come into being? The prevailing theory, beautifully articulated by Lynn Margulies, is that of endosymbiosis, a benevolent act of cellular incorporation, where one prokaryote engulfed another, not in an act of predation, but in a symbiotic embrace that proved mutually beneficial. Consider the mitochondria, the powerhouses of nearly all eukaryotic cells, and the chloroplasts, the photosynthetic engines of plants and algae. These organelles possess their own distinct DNA, replicate independently, and bear striking resemblances to free-living bacteria. It's as if ancient bacterial guests, once independent entities, became permanent residents, their fates intertwined with their hosts. But here, dear friends, the mystery deepens. While the evidence for endosymbiosis is compelling, the precise sequence of events, the identity of the original host cell, and the exact selective pressures that drove this extraordinary partnership remain subjects of intense scientific inquiry. Was the host an archaeon, a bacterium, or perhaps a unique lineage now lost to time? What was the initial advantage that allowed these cellular mergers to not only persist, but to flourish, leading to the emergence of all complex life we see today? It's a story of ancient compromises and profound innovation, a dance of chemistry and energy that still whispers its secrets from within our very cells. From the quiet revolution within the eukaryotic cell, life was poised for its next grand metamorphosis, the advent of multicellularity. For eons, single cells had navigated their existence independently, self-sufficient and autonomous. But imagine the sheer audacity of cells deciding to forgo their solitary ways, to bind together, to specialize, and to cooperate for a greater collective good. This transition from individual cellular autonomy to a coordinated, interdependent cellular society is another profound enigma in the unfolding of life. It's not a single event, but a recurring theme, having evolved independently dozens of times across the tree of life, giving rise to everything from filamentous algae and fungi to the majestic complexity of plants and animals. What were the evolutionary pressures, the silent invitations that encouraged cells to aggregate, adhere, and eventually differentiate into specialized tissues and organs? Was it a quest for increased size, offering protection against predation or better access to resources? Was it the efficiency gained through the division of labor, where some cells focused on reproduction, others on nutrient acquisition, and still others on structural support? The journey from a simple colonial aggregate, where cells merely clump together, to a truly multicellular organism, where cells communicate, cooperate, and sacrifice for the whole, involves a cascade of genetic and developmental innovations. How did the regulatory networks evolve to coordinate such intricate cellular behaviors? How did cells learn to quote for to each other, to send and receive signals that orchestrated growth, development, and repair? We are still deciphering the ancient genetic blueprints and the environmental cues that repeatedly nudged life towards this path of collaborative complexity, a testament to the power of unity in the face of evolutionary challenges. And then, approximately 540 million years ago, after billions of years of microbial dominance and the gradual emergence of simple, multicellular forms, the stage was set for one of the most dramatic acts in the history of life, the Cambrian explosion. For a relatively brief geological span of about 20 to 25 million years, the fossil record suddenly bursts forth with an astonishing array of complex large-bodied animals, many possessing hard parts like shells and exoskeletons, representing nearly all of the major animal phyla we recognize today. Before this period, the fossil record is sparse, dominated by simpler, soft-bodied creatures. After, the oceans teem with trilobites, mollusks, arthropods, and the earliest chordates, the ancestors of vertebrates. This sudden profusion of diverse body plans, this veritable, quote, "five" of morphological innovation, remains one of biology's most captivating and hotly debated unsolved questions. What ignited this fuse? Was it a confluence of environmental factors, perhaps a significant increase in atmospheric oxygen levels that allowed for larger, more metabolically demanding organisms? Did the aftermath of a snowball Earth event, where the planet was largely encased in ice, create new ecological opportunities upon its thaw? Or perhaps the development of new developmental toolkits, genes like the HOX genes, which control body patterning, unlocked an unprecedented capacity for morphological novelty? Some theories suggest a complex interplay, a dynamic feedback loop between predator and prey, where the emergence of efficient predators drove an arms race for defensive adaptations, such as shells and rapid movement, leading to accelerated diversification. Others propose that the Cambrian explosion was less an abrupt event, and more a rapid acceleration of processes already underway, with the apparent explosion partly a consequence of better fossilization conditions for organisms with hard parts. Yet, even with these hypotheses, the sheer speed and scale of the Cambrian diversification continue to humble us, reminding us that evolution, while often gradual, can also proceed in breathtaking, transformative bursts, leaving behind profound questions about the triggers and mechanisms of such grand biological innovation. As we ponder these monumental shifts, the emergence of the eukaryotic cell, the leap to multicellularity, and the grand spectacle of the Cambrian explosion, we are continually invited to consider the deeper whys of life's trajectory. Among these, perhaps none is more puzzling, more counterintuitive than the widespread prevalence of sexual reproduction. Why did life, which could so efficiently replicate itself through asexual means, embrace a strategy that is inherently costly? Sexual reproduction requires finding a mate, expends significant energy, and perhaps most perplexing, only passes on half of an organism's genes to its offspring. Asexual organisms, by contrast, can reproduce rapidly and pass on all their genes, seemingly a more efficient strategy for perpetuating a lineage. Yet sex is ubiquitous among eukaryotes, from microscopic protists to towering trees and complex animals. What is its profound evolutionary advantage that outweighs its obvious costs? The most compelling hypothesis centers on genetic variation. Sexual reproduction shuffles genes, creating new combinations with each generation, a powerful engine for adaptability in ever-changing environments. It allows beneficial mutations to be combined and deleterious ones to be purged more effectively. It provides a defense against parasites and pathogens, as a genetically diverse population presents a moving target, constantly evolving new resistances. But even with these explanations, the precise mechanisms by which sexual reproduction arose, its initial selective advantages in the earliest eukaryotes, and the ongoing evolutionary pressures that maintain its prevalence, continue to be areas of active research and spirited debate. It is a testament to the elegant, often paradoxical solutions that life devises in its relentless pursuit of survival and propagation. We have traversed billions of years in this chapter, from the enduring mystery of Luca's precise form to the grand architectural marvel of the eukaryotic cell, from the collaborative genius of multicellularity to the dazzling spectacle of the Cambrian explosion, and into the fundamental enigma of sexual reproduction. Each step on this evolutionary journey has presented not just a historical event, but a profound invitation to wonder, a series of unsolved questions that beckon us to look closer, to think deeper. The unfurling tapestry of life is rich with such threads of mystery, each one a testament to the immense creativity of natural selection and the limits of our current understanding. As we conclude this exploration of life's early diversification, we are left with a renewed sense of awe for the sheer ingenuity of biology, and a humble appreciation for the vast expanses of knowledge yet to be charted. We have seen life transition from a single spark to a complex, interacting web. But what about the intricate mechanisms within these living forms? How do they maintain their delicate internal balance, respond to their environments, and ultimately age and perish? Our next journey together will lead us into the very heart of these questions, into the intricate dance of homeostasis and the profound mysteries of aging and death. We have journeyed through the initial spark of life and traced its intricate branching into the magnificent diversity we witness today, marveling at the colossal leaps from single cells to complex organisms, from simple replication to the profound enigma of sexual reproduction. Now, as the soft light of our intellectual dawn deepens into a contemplative morning, we turn our gaze inward, towards the very essence of what it means for a living entity to persist, to thrive, and ultimately to yield. We are about to explore the profound ballet of internal balance, the relentless march of time upon biological forms, and the ultimate cessation of that intricate dance, homeostasis, aging, and death. Consider, if you will, the tireless efforts of a cell, an organ, an entire organism to maintain its internal equilibrium amidst a world of constant flux. This unwavering pursuit, this dynamic constancy, is what we call homeostasis. It is life's quiet, steadfast promise to itself to keep its temperature within a narrow range, its pH exquisitely balanced, its glucose levels precisely calibrated, its water content rigorously managed. Imagine a master conductor leading an orchestra of countless biochemical reactions, each note, each crescendo and diminuendo, perfectly synchronized to sustain the melody of existence. That, in essence, is the marvel of homeostasis. How did such an astonishing capacity for self-regulation arise? From the earliest protocells, battling the chaotic chemistry of the primordial soup, to the complex mammals regulating their body temperature with exquisite precision, the ability to maintain internal stability has been non-negotiable for survival. We understand many of the mechanisms, the intricate feedback loops, both negative and positive, that constantly monitor, adjust and correct. A rise in body temperature triggers sweating and vasodilation. A drop activates shivering and vasoconstriction. Blood sugar levels are meticulously controlled by hormones like insulin and glucagon, orchestrating the uptake and release of glucose. But how did these systems first emerge in such rudimentary forms, eventually evolving into the finely tuned, multi-layered regulatory networks we observe across all kingdoms of life? What were the evolutionary pressures that sculpted such exquisite sensitivity and responsiveness? It is not merely a matter of survival, but of thriving, of extending the reach of life into every conceivable niche. From the deepest ocean vents to the highest mountain peaks, each organism a testament to the power of internal resilience. The question remains, how did chemistry learn to self-govern with such an intelligent, almost prescient grace? How did the raw materials of existence weave themselves into such a responsive, self-correcting tapestry? Yet, even the most perfectly tuned instrument eventually shows signs of wear. The tireless efforts of homeostasis, while remarkable, are not eternal. This brings us to another profound and inescapable truth of life. Aging, or senescence. We witness it everywhere, the wilting leaf, the fading bloom, the slowing gait of an elder. It is the gradual, often imperceptible, deterioration of physiological functions, a slow relinquishing of the body's once robust capacity for self-repair and regulation. Why do we age? Is it an inherent flaw in life's design, or a deliberate programmed process? Evolutionary biology offers compelling, though incomplete, answers. One prominent theory, antagonistic pleiotropy, suggests that genes beneficial early in life, enhancing reproductive success, might have detrimental effects later on, once reproduction has largely concluded. Imagine a gene that boosts fertility in youth, but contributes to cellular wear and tear in old age. Nature, prioritizing the propagation of the species, might favor such a gene. Another perspective, the disposable soma theory, posits that an organism has finite resources. It must allocate these resources between maintaining its body soma and reproducing. From an evolutionary standpoint, it is often more advantageous to invest heavily in reproduction, ensuring the continuation of genetic material, even if it means compromising the long-term maintenance of the individual body. The body, in this view, is somewhat, quote, 11, quote, after its reproductive purpose is served. Delving deeper into the microscopic realm, we find the cellular signatures of aging. Telomeres. The protective caps at the ends of our chromosomes shorten with each cell division, eventually signaling cells to stop dividing or to enter a state of senescence. We see the relentless accumulation of cellular damage, oxidative stress from reactive oxygen species, the misfolding and aggregation of proteins, the gradual decline in the efficiency of mitochondria, the powerhouses of our cells. There are also subtle yet profound epigenetic changes, alterations in gene expression without changes to the underlying DNA sequence, which can disrupt cellular function over time. Each of these mechanisms contributes to the unraveling the intricate order that homeostasis strives to maintain. But is aging truly an unavoidable consequence of living? Or could it be a process susceptible to manipulation, perhaps even reversal? The existence of organisms that exhibit negligible senescence, like certain jellyfish that can revert to an earlier life stage, or turtles and whales that appear to age far slower than humans, challenges our assumptions. These biological outliers whisper possibilities, inviting us to question the inevitability of our own decline. What if aging is not a fixed fate, but a malleable program? This question propels vast fields of research, promising not merely extended lifespans, but extended health spans, allowing us to live more fully for longer. And then we arrive at the ultimate cessation, the profound silence that follows life's vibrant song. Death. It is the final surrender of homeostasis, the moment when the intricate biological order dissolves, and the organism can no longer maintain its internal equilibrium against the relentless forces of entropy. Biologically, death plays a crucial, albeit stark, role. It clears the stage for new generations, preventing overcrowding, recycling resources, and ensuring that only the fittest, most adaptable genetic lines continue. It is an evolutionary imperative, a necessary pruning for the flourishing of life as a whole. We see a form of programmed death even within a healthy organism's lifespan. Apoptosis, or programmed cell death. This is a controlled, deliberate process where cells, having served their purpose or become damaged, gracefully dismantle themselves, preventing inflammation and making way for new, healthy cells. Apoptosis is essential for development, tissue maintenance, and disease prevention. It is a testament to life's foresight, a mechanism of self-sacrifice for the greater good of the organism. But how does this controlled cellular demise relate to the death of the entire organism? What truly defines the moment of death at a biological level? Is it the cessation of brain activity, the heart's final beat, or a more complex, systemic unraveling? And beyond the biological necessity, the evolutionary role, lies the profound philosophical weight of individual death. Is there a quote 12 to our individual cessation beyond population dynamics? How does consciousness, that most elusive of biological phenomena, relate to the physical dissolution of the brain? These are questions that transcend the purely scientific, touching upon the very core of our existence and our understanding of what it means to be alive. Homeostasis, aging, and death are not discrete phenomena but deeply intertwined facets of the same grand biological narrative. Homeostasis is the active, dynamic struggle to maintain life. Aging is the gradual, inevitable failure of that struggle, a slow yielding to entropy. And death is the ultimate final surrender. Together, they form a complete arc, defining the boundaries and the duration of every living story. They remind us that life is precious not only for its vibrancy, but also for its impermanence, a fleeting, beautiful defiance against the universal tendency towards disorder. As we conclude this chapter, our minds perhaps a little heavier with these profound truths, we are reminded of the exquisite fragility and astonishing resilience inherent in all biological forms. These are not merely academic questions. They are invitations to wonder at the very fabric of our being, at the universal laws that govern our existence and our eventual return to the dust from which we came. But even in this contemplation of endings, there is a beginning. For in understanding death, we gain a deeper appreciation for life, and in questioning its mechanisms, we push the boundaries of our knowledge. Our journey through the dreamy academy continues, and as the subtle currents of thought guide us forward, we shall next turn our attention to perhaps the most complex and personal of all biological mysteries, consciousness itself and the intricate workings of the brain. As the echoes of our reflections on death gently recede, leaving behind a profound stillness, we find ourselves drawn inexorably toward another magnificent, bewildering frontier, the inner universe of the mind itself. If death marks the cessation of life's intricate dance, then consciousness, that luminous, subjective experience, is the very music that animates it, the silent symphony played within the chambers of our being. We have contemplated the spark of life, its blossoming diversity, and its eventual fading. Now we turn our gaze inward, toward the most astonishing and enigmatic product of biological evolution, the human brain and the mysteries it holds. Imagine, if you will, drifting through a cosmic observatory, not of distant stars, but of the neural constellations within our own skulls. Here, within this soft, convoluted mass resides the capacity for love, for art, for science, for sorrow, and for the very act of questioning our existence. It is by far the most complex object we know in the universe, a universe unto itself, housing perhaps 86 billion neurons, each connected to thousands of others, forming a network of staggering intricacy. Yet, despite millennia of philosophical inquiry and centuries of scientific investigation, the fundamental question persists. How does this exquisitely organized biological machinery give rise to the rich tapestry of subjective experience we call consciousness? This is often termed the hard problem of consciousness, a phrase coined by philosopher David Chalmers, and it stands as one of biology's most formidable challenges. We can, with increasing precision, map the brain's activity, identifying the neural correlates of consciousness, specific patterns of brain function associated with particular experiences. We can observe which areas light up when you see a red apple, feel a pang of sadness, or recall a childhood memory. These are the easy problems concerning the mechanics and functions of the brain. But the hard problem asks, why does any of this physical activity feel like anything at all? Why isn't it all just electrochemical reactions, devoid of the inner light of subjective experience, of qualia, the redness of red, the sweetness of sugar, the ache of loss? Is consciousness an emergent property, something that arises spontaneously from the sheer complexity of neural interactions, much as wetness emerges from countless water molecules, though no single molecule is wet? Or is it something more fundamental, a property woven into the very fabric of reality, waiting for the right biological conditions to manifest? Theories abound, from the integrated information theory, which attempts to quantify consciousness based on a system's capacity for integrated information, to quantum theories that propose a role for subatomic processes within neurons. Yet each offers but a glimmer, a partial lens through which to view an ocean of unknowing. We stand at the shore, sensing the immense depth, but unable to plumb its ultimate secrets. From the grand mystery of consciousness, we descend into its foundational pillars, beginning with the astonishing phenomenon of memory. What intricate architecture allows us to retain fragments of our past, shaping our present and informing our future? It is not, we now understand, a simple recording device, a pristine archive of every moment. Rather, memory is a dynamic, reconstructive process, a living narrative constantly being edited, embellished, and sometimes even invented. Think of a memory not as a fixed photograph, but as a path worn through a dense forest. Each time you traverse that path, it becomes a little clearer, a little easier to navigate. In the brain, this is governed by synaptic plasticity, the remarkable ability of the connections between neurons called synapses to strengthen or weaken over time. When you learn something new or recall an old experience, specific neural circuits are activated, and the synapses within those circuits are modified, making it easier for the same pattern of activity to be reignited in the future. This is how short-term memories, fleeting and fragile, can be consolidated into long-term memories, seemingly etched into our neural landscape. But where, precisely, do these memories reside? It is not in a single, dedicated chamber, but rather distributed across vast networks of neurons, woven into the very fabric of our cerebral cortex, with different brain regions specializing in different aspects. The hippocampus, for instance, acts like a temporary staging area, crucial for forming new declarative memories, facts and events, before they are gradually transferred to the cortex for long-term storage. Emotional memories, often vivid and potent, are deeply intertwined with the amygdala, and procedural memories, the unconscious knowledge of how to ride a bicycle or play a musical instrument, live in different circuits altogether, often involving the cerebellum and basal ganglia. The sheer elegance of this distributed system, its capacity for both incredible detail and profound generalization, is nothing short of miraculous. Yet, the precise mechanisms by which these countless synaptic changes coalesce into a coherent, subjective recollection, complete with sensory details and emotional resonance, remain a profound puzzle. Why do some memories endure with crystalline clarity, while others fade into an irretrievable haze? What dictates the selective tapestry of our personal histories? Closely intertwined with memory is the equally profound mystery of learning. How does our brain, this extraordinary biological computer, adapt, acquire new skills, and continuously reshape itself in response to experience? This capacity for neuroplasticity is perhaps the brain's most defining feature, allowing us to transcend rigid genetic programming and thrive in an ever-changing world. From the simplest forms of classical conditioning to the mastery of complex languages or abstract mathematics, learning represents a fundamental reshaping of our neural architecture. At its core, learning involves the modification of those same synaptic connections we discussed. When two neurons repeatedly fire together, their connection strengthens, a principle often summarized as, quote, 16. This Hebbian plasticity allows the brain to form associations, build neural circuits for specific tasks, and refine its responses over time. But the process is far more nuanced. It involves the growth of new synapses, the pruning of old ones, and even the birth of entirely new neurons in certain brain regions, a process called neurogenesis, challenging older dogmas that the adult brain was fixed. Consider the development of a child, a period of explosive learning. From recognizing faces and voices to acquiring language and motor skills, the young brain is a crucible of plasticity, rapidly forming and reinforcing connections. There are 17 in development where the brain is exceptionally receptive to certain types of input, such as language acquisition or visual processing. But even in adulthood, though plasticity may be less dramatic, it never truly ceases. We are, in essence, lifelong learners, our brains constantly being sculpted by every new experience, every thought, every interaction. And what role does our genetic inheritance play in this? While our genes provide the blueprint for the brain's initial structure, they do not dictate every connection. Epigenetics. The study of how gene expression can be modified without altering the underlying DNA sequence offers tantalizing clues. Environmental factors, experiences, and even diet can influence which genes are turned on or off in our neurons, subtly shaping our neural circuits and influencing our capacity for learning and memory. This dynamic interplay between nature and nurture, between our inherited predispositions and the lifelong symphony of our experiences, adds another layer of breathtaking complexity to the mystery of how we become who we are. These profound questions regarding consciousness, memory, and learning inevitably lead us to the very concept of the self, and the enduring debate surrounding free will. If our thoughts, emotions, and decisions are merely the product of electrochemical reactions within our neural networks, are we truly the authors of our own choices? Is the self an illusion, a compelling narrative spun by our brains to create a sense of coherence amidst the constant flux of sensory input and internal processing? Neuroscientific experiments demonstrating that brain activity associated with a decision can precede our conscious awareness of making that decision, challenge our deeply held intuitions about agency. Yet, the subjective experience of making a choice, of having intentions, of being a distinct individual navigating the world is undeniable. This tension between the deterministic dance of neurons and the felt reality of personal freedom remains one of the most profound philosophical and scientific conundrums, inviting us to ponder the very nature of what it means to be human. Is our sense of self merely an intricate pattern of information, an emergent property that gives rise to a compelling illusion of singularity? Or is there something more, something that transcends the purely physical, residing in the very heart of the qualia we experience? As we peer into these depths, the future beckons with both promise and trepidation. Ambitious projects like the Human Connectome project aim to map the entire network of neural connections within the human brain, offering an unprecedented level of detail about its architecture. Advances in artificial intelligence and machine learning are allowing us to build models that mimic certain aspects of brain function, raising questions about the potential for artificial consciousness. Yet, even as we make strides in understanding the brain's mechanics, the quote, 20 of consciousness the subjective what-it-is-likeness of experience remains stubbornly elusive. We stand, then, at the threshold of our own minds, gazing into a mirror that reflects the universe's grandest mystery. The brain, this 3-pound universe of thought and feeling, continues to guard its most profound secrets, offering us not answers, but invitations to wonder. We have only begun to chart its vast landscapes, to decipher the language of its neurons, and to truly comprehend the miracle of our own sentience. The journey inward is perhaps the longest and most challenging of all, promising revelations that will undoubtedly reshape our understanding of life itself, and perhaps even our place within the cosmos. As our exploration of these internal landscapes concludes for now, we will next venture outward once more to consider how these incredibly complex minds and the organisms they inhabit interact with the world around them, shaping and being shaped by the intricate webs of ecology and evolution. As we gently drift away from the profound, introspective landscapes of consciousness and the elusive self, where the very essence of our individual experience resides, we find ourselves drawn to another boundless frontier, one that exists not in the vastness of the cosmos, nor solely within the intricate folds of our own brains, but rather within and around us, a hidden universe teeming with life. We now turn our gaze together to the unseen architects of our existence, the silent partners in our biological narrative, the microbiome. Imagine, if you will, that the self we so carefully contemplate is not a singular, isolated entity, but rather a vibrant, bustling metropolis, a living ecosystem composed of countless inhabitants. This is the truth of our being, a truth that science is only now beginning to fully apprehend. We are not merely human. We are intricate symphonies of human cells and microbial cells, a tapestry woven from the threads of countless species. For every cell that bears our unique genetic signature, there are, by some estimates, at least as many, if not more, microbial cells, bacteria, archaea, fungi, viruses, and protozoa cohabiting our bodies, particularly within the labyrinthine passages of our gut, upon the expansive canvas of our ski, and within the delicate ecosystems of our oral and urogenital tracts. This collective community of microorganisms, along with their genes and metabolites, is what we term the microbiome. It is a concept that has revolutionized our understanding of biology, transforming our view of the individual organism from a solitary fortress to a dynamic, interconnected biome. For centuries, our focus in medicine and biology was predominantly on the human host, often viewing microbes as mere invaders or passive inhabitants. But now we are learning to appreciate them as indispensable partners, co-evolutionary companions whose presence and activity are fundamental to our health, our development, and perhaps even our very identity. The sheer scale of this microbial universe is an initial invitation to wonder, is it not? Consider the human gut microbiome alone, a teeming metropolis within our intestines. Here, billions upon billions of microorganisms engage in a constant, intricate dance of metabolism, communication, and competition. They break down complex carbohydrates that our own enzymes cannot digest, synthesizing essential vitamins, modulating our immune system, and even influencing the delicate balance of our neural chemistry. To consider this vast internal world is to confront a fundamental question. How did this profound symbiosis evolve, and how is its delicate equilibrium maintained, often without our conscious awareness? One of the most compelling mysteries surrounding the microbiome begins at the very dawn of an individual's life. How is this initial microbial community established, this foundational blueprint that will likely influence a lifetime of health? We understand that the first significant colonization occurs during birth, as a newborn traverses the maternal birth canal, acquiring a unique microbial legacy. But what are the precise ecological succession events that follow? How do factors like mode of delivery, early diet, breast milk versus formula, and environmental exposures shape this nascent ecosystem? Is there an optimal quote 22 process, a specific sequence of microbial arrivals that lays the groundwork for lifelong resilience? And what are the long-term consequences, both subtle and profound, of deviations from these initial pathways, particularly in an era of increasing C-sections and sterile environments? These questions hint at a delicate developmental window, a period of immense plasticity, where the future health trajectory of an individual might be subtly, yet powerfully determined by their unseen microbial companions. Beyond its genesis, we grapple with defining what constitutes a microbiome. Is there a universal template, a canonical composition that all thriving human beings share? Or is health defined by a personalized ecology, a unique microbial fingerprint adapted to an individual's genetics, diet, lifestyle, and environment? The emerging evidence suggests the latter. A remarkable diversity exists even among healthy individuals, defying a one-size-fits-all definition. Instead, perhaps, health in this context refers to functional resilience, the ability of the microbial community to resist perturbations, to recover from disturbances, and to perform its vital functions efficiently, irrespective of the precise species composition. But what are the underlying ecological principles that govern this resilience? How do complex networks of thousands of species cooperate and compete to maintain stability in the face of antibiotic challenges, dietary shifts, or environmental stresses? The answers lie hidden in the intricate web of microbial interactions, a biological puzzle of unparalleled complexity. Perhaps the most astonishing and currently unresolved frontier of microbiome research lies in its profound influence on host physiology and behavior. We are increasingly aware of the gut-brain axis, a bidirectional communication highway linking the enteric nervous system, the central nervous system, and the gut microbiome. But how, precisely, do these microbial inhabitants, lacking neurons themselves, orchestrate changes in our mood, cognition, and susceptibility to neurological disorders? We know they produce a vast array of neuroactive compounds, including short-chain fatty acids, neurotransmitter precursors, and even direct neurotransmitters like serotonin and GABA. But how do these molecular messages traverse the gut barrier, navigate the bloodstream, cross the blood-brain barrier, and ultimately modulate the intricate symphony of our neural networks? Are they direct signals, or do they act indirectly, perhaps by influencing the immune system, which in turn communicates with the brain? The tantalizing links between gut dysbiosis and conditions like anxiety, depression, autism spectrum disorders, and even neurodegenerative diseases like Parkinson's suggest a profound yet still largely mysterious dialogue unfolding within us. Consider, too, the microbiome's intimate dance with our immune system. From the moment of birth, our commensal microbes act as vital educators, shaping the development and maturation of our immune defenses. They teach our immune cells to distinguish between friend and foe, to tolerate harmless antigens, and to mount appropriate responses against pathogens. How do they achieve this sophisticated immunological tutoring? What are the specific microbial components or metabolites that interact with immune receptors, triggering cascades of signaling pathways that fine-tune our inflammatory responses? The rise of allergies, autoimmune diseases, and inflammatory bowel conditions in modern societies, often linked to altered microbial exposure, underscores the critical, yet still poorly understood role of the microbiome in immune homeostasis. Unraveling these molecular dialogues holds the promise of entirely new therapeutic strategies for a spectrum of chronic illnesses. And what of metabolism and nutrition? Our microbial partners are veritable chemical factories, extracting energy and nutrients from food components that our own bodies cannot process. They synthesize vitamins, modify bile acids, and produce a plethora of metabolites that influence our energy balance, satiety, and susceptibility to metabolic diseases like obesity and type 2 diabetes. But the precise mechanisms by which specific microbial consortia influence these outcomes remain largely elusive. Why do some individuals gain weight more readily than others on similar diets? And what role do their unique microbial communities play in this variability? Can we truly engineer a microbiome to optimize metabolism, or are we merely scratching the surface of an incredibly complex, personalized metabolic interplay? The mysteries extend beyond the gut, encompassing the distinct microbiomes of our skin, mouth, lungs, and other body sites. Each represents a unique ecological niche with its own set of unresolved questions about community assembly, functional contributions, and influence on local and systemic health. The skin microbiome, for instance, plays a crucial role in barrier function and immune defense. Yet how its diverse inhabitants maintain balance and prevent dermatological conditions is still an active area of inquiry. As we ponder these intricate relationships, we are led to a profound realization. We are not merely hosts to these microbes. We are, in a very real sense, co-evolved ecosystems. Our evolutionary journey has been inextricably intertwined with theirs, a grand biological partnership forged over eons. This perspective challenges our anthropocentric view, inviting us to acknowledge our profound interdependence and to embrace a more holistic understanding of life. We are living proof that cooperation, even at the microbial level, can lead to astonishing complexity and resilience. The future then holds immense promise and equally immense challenges. The burgeoning field of microbiome therapeutics, from precisely targeted probiotics and prebiotics to the more radical yet effective fecal microbiota transplants, offers tantalizing glimpses of new ways to treat disease and promote health. Yet, without a deeper understanding of the fundamental ecological principles governing these complex communities, our interventions remain largely empirical. We are like gardeners attempting to cultivate a vast, intricate rainforest, with only a rudimentary knowledge of its soil composition, rainfall patterns, or interdependent species. How do we move from correlation to causation? How do we predict the long-term consequences of manipulating such delicate ecosystems? These are not mere scientific questions, but invitations to navigate a new ethical landscape, where the very definition of human health expands to encompass the well-being of our microbial selves. And so, as the twilight deepens around our virtual academy, and we contemplate the vast unseen world within us, we are reminded once more of the boundless interconnectedness of life. The human microbiome is but a microcosm of a larger planetary truth, that all life on Earth, from the smallest bacterium to the largest whale, is part of an immense, self-regulating biological system. The unresolved questions of our own internal microbial ecosystems echo the grander mysteries of how the Earth's global microbiome, from its oceans to its soils to its atmosphere, sustains the very conditions for life. It is a humbling thought, a quiet reminder that our journey through these profound biological enigmas is not merely an intellectual pursuit, but a deepening of our reverence for the intricate, beautiful, and endlessly mysterious fabric of existence itself. Next, as we continue our shared intellectual voyage, we will venture into an entirely different realm of biological mystery, one that challenges our notions of individuality and collective intelligence. The intricate and often baffling world of collective behavior and biological cooperation, from the swarming of insects to the complex societies of social animals, and the unresolved questions of how decentralized decision-making can lead to such emergent complexity. As we gently drifted through the intricate chambers of the microbiome, witnessing the profound, unseen symphony orchestrated by countless microbial lives within us, a new question begins to unfurl, like a delicate blossom opening its petals to the dawn. If our very selves are a tapestry woven from human and microbial threads, a collective of interdependent entities, then how much further does this principle of cooperation extend? What happens when individual organisms, distinct and self-sustaining, choose to intertwine their fates, to sacrifice for a greater whole, to forge something grander than any single entity could ever achieve? Today, we embark upon the exploration of collective behavior and biological cooperation, a realm where the, quote, 26 dissolves into a magnificent, quote, 27, where the boundaries between individual and community blur, and where life's most astonishing feats are often accomplished not by singular heroes, but by synchronized multitudes. We are stepping beyond the confines of a single organism, beyond the internal dialogues of cells and microbes, to observe the breathtaking choreography that unfolds when individuals driven by forces both seen and unseen unite. It is here, at the crossroads of individuality and community, that some of biology's most profound and beautiful mysteries reside. Consider, if you will, the very foundation of your own existence, the multicellular organism. Long ago in the ancient oceans, single-celled life forms began to aggregate, to specialize, to surrender a measure of their autonomy for the collective benefit of a larger, more complex entity. This, as we briefly touched upon in our journey through diversification, was an act of profound biological cooperation, a primordial pact where cells learned to communicate, to coordinate, to even sacrifice themselves through apoptosis for the health and integrity of the whole. Each cell, a tiny citizen, performs its specialized role, contributing to the grand architecture of a tissue, an organ, a complete being. How did this initial leap of faith occur, this fundamental agreement to relinquish individual immortality for the promise of a shared, more complex existence? The whispers of these ancient negotiations still echo in the intricate regulatory networks that govern our bodies, a testament to the enduring power of cooperation. But the dance of cooperation extends far beyond the confines of a single body. Look to the humble bacteria, those ancient architects of life, and you will find them engaging in sophisticated collective behaviors. Think of biofilms, those slimy, resilient communities where microbes of various species adhere to surfaces, encasing themselves in a self-produced polymeric matrix. Within these microscopic cities, bacteria communicate through chemical signals, a process known as quorum sensing, to coordinate gene expression, share resources, and collectively resist threats, displaying a unified front far more potent than any individual could muster. How do these simple life forms orchestrate such complex, coordinated defenses and resource allocation strategies? What are the precise rules governing their communal decision-making, allowing them to transform from independent swimmers into a formidable, collective fortress? Perhaps the most iconic examples of biological cooperation emerge from the insect world, where the concept of the superorganism takes breathtaking form. Consider the ant colony, the beehive, or the termite mound. Vast, intricate societies where tens of thousands, even millions of individuals operate with such seamless coordination that the entire colony functions as a single, cohesive entity. Here, an extreme division of labour prevails. Queens dedicated solely to reproduction, sterile workers performing a myriad of tasks from foraging to nursing to defence, and soldiers specialised for combat. Communication flows through an elaborate language of chemical pheromones, tactile signals and intricate dances, guiding every action from nest construction to complex hunting strategies. How did such profound altruism, the complete surrender of individual reproductive potential for the good of the colony, evolve? What are the genetic and epigenetic mechanisms that hardwire these roles, ensuring that each individual understands and fulfills its part in the collective endeavour? The superorganism challenges our very definition of an individual, prompting us to ponder where the true double quote, 28 double quote, resides in such an intricately interwoven community. Beyond these structured societies, cooperation manifests in more fluid, ephemeral forms. Witness the mesmerizing spectacle of a flock of starlings swirling in the twilight sky, painting dynamic patterns against the canvas of the heavens, or a school of fish darting as one, a shimmering silver organism moving with impossible grace and speed to evade a predator. Consider the terrifying unity of a locust swarm, a collective entity that devours everything in its path. In these instances, there is no central leader, no conductor orchestrating the symphony. Instead, complex emergent behaviors arise from surprisingly simple rules, each individual reacting to its immediate neighbours, maintaining a certain distance, aligning its movement, and avoiding collisions. How did these decentralized systems achieve such astonishing coherence and efficiency? What are the neurological and sensory mechanisms that allow for such rapid, almost telepathic coordination across vast numbers of individuals? The emergent intelligence of these swarms and flocks offers profound insights into self-organization, reminding us that complexity can arise from simplicity, and order can bloom from apparent chaos. Yet, perhaps the greatest enigma embedded within the heart of biological cooperation is the paradox of altruism. In a world governed by natural selection, where the fittest individuals are those most successful at surviving and reproducing, why would any organism engage in behaviours that reduce its own fitness for the benefit of another? Why would a worker bee sting an intruder, sacrificing its own life to protect the hive. Why would a parent bird risk its life to defend its young? Why would a vampire bat share its regurgitated blood meal with a hungry, unrelated roost mate? These acts of self-sacrifice, seemingly counter to the ruthless logic of survival, have long puzzled evolutionary biologists. Over time, several compelling hypotheses have emerged to illuminate this profound mystery. Kin selection, proposed by W. D. Hamilton, suggests that altruistic behavior can evolve if it benefits genetic relatives, thereby ensuring the propagation of shared genes. An individual may perish, but if its sacrifice saves several close relatives, thus, quote 29, the sum of an individual's own reproductive success and the reproductive success of its relatives can be enhanced. It's a subtle, yet powerful redefinition of evolutionary success, extending beyond the individual to the genetic lineage. Then there is reciprocal altruism, where individuals cooperate with the expectation of future repayment, even if that repayment is not immediate. The principle, often modelled using concepts from game theory like the prisoner's dilemma, suggests that repeated interactions can foster a climate where cooperation, rather than defection, becomes the most advantageous long-term strategy. And while more controversial, the concept of group selection posits that groups composed of more cooperative individuals might outcompete groups with fewer cooperators, leading to the spread of altruistic traits. Each of these theories offers a lens through which to view the intricate calculus of cooperation, but the full picture, the precise weighting of these forces in different contexts, remains a vibrant area of inquiry. Further complicating this beautiful tapestry of cooperation is the ever-present shadow of cheaters. If cooperation offers such profound benefits, what prevents individuals from exploiting the system, from reaping the rewards without contributing their fair share? Evolution, in its infinite wisdom, has devised mechanisms to police these systems, to identify and punish those who would undermine the collective good. In bacterial biofilms, chemical signals can detect non-cooperators. In social insect colonies, workers may actively suppress the reproduction of other workers. In human societies, our complex systems of morality, law, and social norms are, in part, elaborate mechanisms to enforce cooperation and deter defection. This constant evolutionary arms race between cooperation and exploitation, between the collective good and individual gain, is a dynamic force shaping the intricate balance of biological systems, a testament to the enduring tension at the heart of life. And what of our own species, Homo sapiens? We stand as perhaps the most intensely cooperative creatures on Earth, capable of organizing into vast societies of millions, even billions, of unrelated individuals, collaborating on projects of immense scale and complexity, from building cities to exploring the cosmos. Our cooperation is not merely driven by genetic relatedness or immediate reciprocity. It is deeply intertwined with culture, language, shared beliefs, and abstract concepts of morality and justice. How did our species cross the threshold from small, kin-based groups to these sprawling, anonymous collectives? What are the unique cognitive and emotional capacities that allow us to trust strangers, to form alliances beyond familial ties, to sacrifice for ideals? The origins of human morality, our capacity for empathy, and our propensity for large-scale, often anonymous altruism remain some of the most profound and humbling questions at the intersection of biology, psychology, and philosophy. As we conclude this contemplation of collective behaviour, we are left not with definitive answers, but with a renewed sense of wonder at the intricate ways life finds to connect, to collaborate, to build something greater than the sum of its parts. How do these collective intelligences process information, make decisions, and adapt to changing environments with such efficiency? What are the genetic and epigenetic switches that allow for such profound shifts from individual autonomy to collective synergy? How do the simple rules governing a flock of birds scale up to the complex, multi-layered cooperation of a human society? The answers lie hidden in the whispers of the wind through a murmuration, in the silent communication within a biofilm, in the ancient pacts forged between the first cells. They are invitations to wonder, to delve deeper into the very fabric of existence, revealing that life, in its essence, is a grand, unfolding story of connection. Next, our journey will take us into an even more intimate and perhaps perplexing realm, the intricate, often invisible world of biological communication and sensory perception. How do organisms truly talk to each other? And how do they construct their unique realities from the symphony of signals that surrounds them? Prepare to explore the unseen languages of life itself, as we journeyed through the intricate landscapes of life, from the spark of abiogenesis to the grand ballets of collective behaviour. We have consistently found ourselves drawn to the underlying threads that weave these phenomena together. We've marvelled at the emergent intelligence of a murmuration of starlings, the collaborative spirit within a bacterial biofilm, and the profound, often sacrificial acts of altruism that bind species together. These complex orchestrations, these symphonies of cooperation, demand a conductor, a score, a blueprint, an underlying system of information that guides each player, each cell, each organism in its assigned role. In our previous explorations, we touched upon the foundational role of DNA, that magnificent double helix, often hailed as the immutable blueprint of life. We learned of its elegant structure, its precise sequence of nucleotides encoding the vast libraries of proteins that build and operate every living thing. It is, we have understood, the very language of heredity, passed with meticulous fidelity from one generation to the next, dictating the colour of our eyes, the shape of our leaves, the very architecture of our being. Yet, as with any grand narrative, the more closely we examine it, the more layers of untold stories, of whispered secrets, begin to unfurl. Today, we turn our gaze to these subtler narratives, to the annotations and interpretations of that fundamental score, to the dynamic interplay between the fixed genetic text and the ever-changing symphony of life itself. We are stepping into the realm of epigenetics, a field that invites us to reconsider the very nature of biological inheritance and the fluid boundaries of our genetic destiny. Imagine, if you will, the genome not merely as a static blueprint etched in stone, but as a dynamic, living manuscript, constantly being read, highlighted, annotated, and even re-edited by the very experiences of life. And it's as if the grand library of our genetic code, while retaining its core texts, possesses a myriad of sticky notes, bookmarks, and marginalia, all influencing how those texts are interpreted and when they are read. These annotations, these subtle modifications that don't alter the underlying DNA sequence itself, but profoundly impact gene expression, are the essence of epigenetics. They are the whispers beyond the code, the environmental echoes inscribed onto our very being. How does this intricate system operate? At its heart lie several elegant mechanisms, each a testament to life's ingenious capacity for regulation. One of the most well-studied is DNA methylation, a process where a small chemical tag, a methyl group, is added to specific cytosine bases within the DNA strand. Think of it as a dimmer switch, often turning down or even silencing the expression of a nearby gene. When a gene is heavily methylated, it's as if a librarian has placed a Do Not Disturb sign on a particular chapter, making it less accessible for reading. Conversely, removing these methyl tags can flip the switch back on, allowing genes to be actively transcribed into proteins. Another crucial player in this dynamic drama is histone modification. Our DNA, an incredibly long molecule, is not simply free-floating within the cell's nucleus. It is meticulously wound around spool-like proteins called histones. This packaging is not merely for organizational purposes, it's a critical regulatory mechanism. When histones are modified, perhaps by the addition of acetyl groups, phosphate groups, or even methyl groups, their grip on the DNA can either loosen or tighten. Picture the DNA as a delicate thread wrapped around spools. If the spools are tightly wound, the thread, and thus the genes it contains, is inaccessible to the cellular machinery that reads it. But if the spools relax their grip, the thread unfurls, making the genes readily available for expression. These modifications therefore act as gatekeepers, controlling the accessibility of our genetic library. And then there are the enigmatic non-coding RNAs, molecules of RNA that, unlike messenger RNA, do not directly translate into proteins. Instead, they play diverse and sophisticated regulatory roles, acting as molecular maestros that can fine-tune gene expression, silence specific genes, or even orchestrate complex epigenetic landscapes. They are these subtle conductors, guiding the cellular orchestra with precision and nuance. What makes epigenetics such a profound and, in many ways, an unsolved mystery is the realization that these epigenetic marks are not merely static, pre-programmed features. They are incredibly dynamic, constantly responding to the world around us, our diet, our stress levels, our exposure to environmental toxins, our social interactions, even our thoughts and emotions. All these can leave their indelible marks on our epigenome. This means that the blueprint of our being is not fixed at conception, but is instead a continuously updated document, a living testament to our ongoing dialogue with the environment. The food we eat today, the sunlight we bask in, the anxieties we carry, these experiences are not just transient events. They are being subtly recorded, translated into biochemical annotations that influence which genes are active and which remain silent within our cells. This brings us to one of the most compelling and controversial unsolved questions in biology: epigenetic inheritance across generations. For decades, the prevailing wisdom, rooted in Mendelian genetics, was that acquired characteristics could not be inherited. Only changes to the DNA sequence in germ cells, sperm and egg, were thought to be passed down. But what if these dynamic epigenetic marks, these environmental annotations, could indeed bridge the generational gap? What if the experiences of a grandparent could, in some subtle yet significant way, influence the gene expression patterns and thus the health and predispositions of their grandchildren? Evidence for such transgenerational epigenetic inheritance is mounting, challenging long-held dogmas and opening breathtaking new avenues of inquiry. In the plant kingdom, it is a well-established phenomenon, allowing offspring to quote 33 and adapt to environmental stresses their ancestors endured. In simpler organisms like nematodes and fruit flies, researchers have observed epigenetic changes induced by diet or stress, persisting for several generations, even in the absence of the original stimulus. The implications for mammals, and especially for humans, are nothing short of revolutionary. Studies on human populations, such as the descendants of those who experienced severe famine, like the Dutch hunger winter, have suggested that epigenetic changes related to metabolism and health risks can be transmitted. Children and even grandchildren of famine survivors have shown altered risks for conditions like diabetes and cardiovascular disease, potentially linked to epigenetic modifications. Similarly, research into the effects of trauma, stress, and even social adversity hints at epigenetic signatures that could be passed down, influencing susceptibility to mental health disorders or stress responses in subsequent generations. Is it possible that the emotional burdens and adaptive strategies of our forebears are not just historical footnotes, but subtle biological legacies encoded not in our genes, but in the way our genes are read? This phenomenon, if widespread and robust in humans, profoundly blurs the long-standing lines between quote 34 and quote 35, suggesting an intricate, continuous conversation rather than a clear dichotomy. It hints at a form of inheritance that is more plastic, more responsive, and perhaps more forgiving or damning than we ever imagined. It suggests that our choices, our environments, and our experiences might not only shape us, but also subtly influence the biological landscapes of our descendants, creating a profound sense of intergenerational connection and responsibility. But how do these epigenetic marks survive the rigorous quote 36 quote that typically occurs during germline development, where most epigenetic tags are supposedly erased to ensure a blank slate for the new embryo? This is a significant part of the unsolved mystery. What are the precise molecular mechanisms that allow some epigenetic information to evade this erasure, to persist, and to be faithfully transmitted? And why would evolution favour such a system? Perhaps it offers a rapid, adaptive mechanism for organisms to prepare their offspring for predictable environmental challenges, allowing for quicker responses than traditional genetic mutation and natural selection. It is a biological foresight, a pre-adaptation, whispered from past generations to future ones. As we stand at the precipice of understanding this dynamic genome, we are invited to ask deeper, more philosophical questions. What does this mean for our concept of self, if our biological identity is not solely defined by the static sequence of our DNA, but also by the ever-changing tapestry of our epigenetic landscape, influenced by every breath we take, every meal we eat, every emotion we feel? Does it mean we are more deeply connected to our environment and to our ancestral past than we ever conceived? And what responsibility does this newfound understanding place upon us, knowing that our present actions might echo through the epigenomes of generations yet to come, The genome, then, is not merely a book, it is a symphony, a living score constantly being interpreted and reinterpreted, annotated by the very act of living. Epigenetics unveils a layer of biological information that is both ancient and profoundly modern, a bridge between our inherited past and our adaptive future. It is a testament to life's incredible ingenuity, its capacity to write new stories on old pages, to find novel ways of transmitting wisdom beyond the confines of the genetic code. As we move forward, we carry with us this expanded understanding, this profound appreciation for the whispers beyond the code, eager to discover what other layers of biological information await our patient and wondering gaze. What other secrets, we ponder, does the dynamic genome still hold, poised to unfurl like a delicate blossom in the quiet dawn of discovery? Having journeyed through the intricate landscapes of epigenetics, where the environment's subtle whispers can alter the very expression of our genes, even across generations, we now turn our gaze to a related yet distinct marvel, the ceaseless symphony of biological communication and the profound mystery of sensory perception. If epigenetics reveals the enduring echoes of ancestral experiences, then communication and perception are the vibrant real-time dialogues that animate every living moment, shaping the intricate dance between organism and environment, between self and other. Imagine, if you will, a vast living tapestry, woven not just from threads of DNA, but from invisible filaments of information, constantly exchanged, received, and interpreted. This, dear friends, is the essence of biological communication. It is a concept far broader than human language, encompassing every signal, every molecule, every gesture that carries meaning between biological entities. From the most ancient single-celled bacterium to the most complex human society, life is an incessant conversation, a grand, multi-layered exchange of data that orchestrates existence itself. At its most fundamental, communication begins within the cell, a bustling metropolis of molecular messengers. How do these tiny, self-contained universes manage their internal affairs with such breathtaking precision? We speak of signaling pathways, cascades of proteins and ions that relay instructions from the cell membrane to the nucleus, orchestrating metabolism, growth, and division. But how does a cell, without a brain or conscious intent, interpret a subtle shift in its internal chemistry as a command to divide or a threat to initiate repair? What is the language of these intracellular dialogues, and how did such an exquisitely complex system of molecular recognition and response first evolve? It is a silent chemical poetry whose grammar we are only beginning to decipher. Then we widen our lens to the intercellular realm, where cells communicate with their neighbours, forming tissues, organs, and ultimately entire organisms. Hormones, those potent chemical couriers, traverse vast distances through the bloodstream, carrying messages of hunger, stress, or passion. Neurotransmitters leap across synaptic clefts, sparking thoughts, emotions, and movements. Growth factors guide development, immune signals coordinate defenses, and countless other molecular emissaries maintain the delicate homeostasis of the body. Yet, even here, profound questions persist. How do cells distinguish between a multitude of competing signals, filtering noise from vital information? What are the precise algorithms that allow a collective of billions of cells to cooperate so seamlessly, avoiding chaos and maintaining the integrity of the whole? The sheer sophistication of this cellular communication network, operating with an efficiency and resilience that dwarfs our most advanced technologies, remains a profound source of wonder. Moving beyond the confines of a single organism, communication unfurls into the vibrant interactions between individuals and species. Consider the silent yet potent language of pheromones, chemical signals that guide insects to mates, warn of predators, or mark territories. Think of the elaborate visual displays of birds in courtship, the intricate dances of honeybees conveying the location of nectar, or the haunting, complex songs of whales that traverse ocean depths. These are not mere behaviours. They are sophisticated acts of communication, encoding vital information about identity, intention, and environment. And what of the communication between species? The warning calls of a bird alerting others to a predator, the mimicry of a harmless species adopting the appearance of a dangerous one, the intricate chemical signals exchanged between plants and their pollinators, or between trees connected by subterranean fungal networks. How did these diverse, often species-specific communication systems evolve? What are the fundamental principles underlying their design, allowing for both specificity and adaptability across the vast spectrum of life? And this brings us to the profound interplay of communication with its indispensable partner, sensory perception. If communication is the act of sending information, perception is the miraculous faculty of receiving and interpreting it, transforming raw stimuli into meaningful experience. Our human senses, sight, sound, touch, taste, and smell, offer us a rich, albeit limited, window into the world. But the biological realm teems with senses far beyond our immediate comprehension, inviting us to contemplate realities we can barely imagine. Consider the astonishing diversity of chemoreception. While we perceive taste and smell, many organisms possess an exquisite sensitivity to chemical gradients, guiding bacteria towards nutrients, or allowing a shark to detect a single drop of blood miles away. What is the subjective experience of a moth navigating by a pheromone plume across kilometers, or a salmon finding its way back to its birth stream by the faint chemical signature of its waters? Mechanoreception, too, extends far beyond our sense of touch and hearing. Fish possess lateral lines, detecting subtle water movements and pressure changes. Spiders feel the world through vibrations in their webs. Plants perceive touch, wind, and gravity, responding with growth patterns that defy our understanding of feeling. How do these diverse mechanosensors translate physical force into biological signals? And how do organisms integrate these myriad pressures into a coherent understanding of their physical surroundings? Photoreception, our vision, is but one facet of light perception. Many insects see in ultraviolet, perceiving patterns on flowers invisible to us. Some deep-sea creatures detect bioluminescence, while pit vipers possess infrared sensing organs, allowing them to see the heat signatures of their prey in absolute darkness. What does the world look like through the eyes of a bee or a snake? Are there fundamental colours and patterns that exist yet remain forever outside our perceptual grasp? And then we delve into truly alien senses. Electroreception, the ability to detect electric fields, allows sharks to find prey hidden in sand, or platypuses to hunt in murky waters. Magnetoreception, the faculty of sensing the Earth's magnetic field, guides migratory birds, sea turtles, and even bacteria across vast distances. How do living cells generate and interpret these incredibly subtle electromagnetic signals? Is it a quantum phenomenon, a biological compass embedded at the atomic level, or something else entirely? The mechanisms remain largely elusive, shrouded in the enigmatic physics of life. The greatest mystery, perhaps, lies not just in how these signals are received and processed, but in the transformation of raw sensory data into subjective experience. This brings us back to the quote of consciousness we explored earlier. How does the firing of neurons triggered by light waves or chemical molecules give rise to the vivid personal sensation of seeing the colour red or tasting the sweetness of honey? How do different sensory inputs, the sight of a predator, the sound of its approach, the scent of its presence, integrate into a single, unified and actionable perception of threat? We are, in essence, asking, what is reality to a bat navigating by echolocation? What is the world like for a plant constantly sensing light, water, nutrients, and the touch of every passing creature, yet without a brain? Each organism, with its unique suite of communicative tools and perceptual filters, constructs its own version of reality, a subjective universe shaped by its evolutionary history and ecological niche. We are but one tiny island of perception in an ocean of countless ways of experiencing existence. As we ponder these profound questions, we are reminded of the inherent humility of scientific inquiry. The universe of biological communication and sensory perception is far richer, more complex and more mysterious than we could ever fully grasp. Each unanswered question is not a void, but an invitation, a beckoning finger pointing towards new frontiers of discovery. We are learning to listen more closely to the whispers of cells, to observe the silent dialogues of ecosystems, and to imagine the myriad ways in which life perceives and interacts with its boundless environment. The journey continues, ever deeper into the heart of what it means to be alive, to connect and to experience. As our journey through the living world deepens, we have collectively marvelled at the intricate dance of communication that binds organisms, at the unique realities each creature sculpts from the raw data of its senses. We have seen how life, in its boundless creativity, perceives and interacts with its environment in myriad, astonishing ways, from the subtle chemical whispers between cells to the grand symphonies of interspecies dialogue. Yet, what if the very stage upon which these dramas unfold, the fundamental fabric of biological existence itself, is woven with threads far finer, far more enigmatic than we have yet fully appreciated? What if, beneath the bustling, seemingly classical world of our cells and senses, there lies a deeper quantum reality, quietly shaping life in ways that defy our conventional understanding? Today, dear companions, we invite you to step with us into an even more profound quietude, a realm where the laws of the very small begin to whisper their secrets to the grand architecture of life. We are venturing into the nascent, yet utterly mesmerizing field of quantum biology, a frontier that challenges our most cherished assumptions about how life works, suggesting that evolution, in its infinite ingenuity, might have learned to harness the strange, counterintuitive powers of the quantum realm. For generations, the domain of biology has largely been understood through the lens of classical physics and chemistry. We have pictured molecules as tiny, hard spheres interacting like billiard balls, and reactions as straightforward collisions. Quantum mechanics, with its ethereal probabilities, its particles existing in multiple states simultaneously, its spooky actions at a distance, seemed confined to the frigid, isolated vacuums of laboratories, a world far removed from the warm, wet, noisy complexity of a living cell. How, we mused, could such delicate quantum phenomena possibly persist, let alone play a functional role, amidst the chaotic thermal vibrations and constant interactions within biological systems? But the whispers have grown louder, the evidence more compelling, suggesting that life is not merely tolerating the quantum realm, but exploiting it. We are beginning to glimpse an unseen architecture, a subtle orchestration where the universe's most fundamental laws are not just background noise, but active participants in life's most essential processes. It is an invitation to wonder, not just at the complexity of life, but at the very elegance of its design, pushing the boundaries of what we thought possible. Consider, if you will, the miracle of curated photosynthesis, the ancient alchemy by which plants, algae, and some bacteria capture sunlight and transform it into the energy that fuels nearly all life on Earth. We know the basic steps. Photons strike chlorophyll molecules, exciting electrons, and this energy is then transferred through a complex of proteins until it can be used to synthesize sugars. But the efficiency of this process, particularly the initial energy transfer, is breathtakingly high, often approaching 95 to 100%. If energy were simply hopping randomly from one molecule to the next, much of it would be lost as heat. Yet, it seems to navigate a dense molecular forest with almost perfect precision. Here, the quantum whisper becomes a resonant chord. Scientists now theorize that the excited energy, or quote 40, within the light harvesting complexes doesn't just hop. It explores multiple pathways simultaneously, much like a quantum particle existing in a superposition of states. This allows it to sample all possible routes to the reaction center and choose the most efficient one, collapsing its wave function upon arrival. It is as if the energy itself possesses an innate quantum GPS, finding the optimal path through a dense network of chromophores. This phenomenon, known as a quantum coherence, where particles maintain a synchronized wave-like state, was thought to be impossible in the warm, bustling environment of a cell. Yet, life seems to have evolved a way to protect and harness it, like a master architect of light guiding its precious energy with unparalleled grace. How did this astonishing ability evolve? What are the precise molecular mechanisms that maintain this fragile coherence, shielding it from the thermal chaos? These remain profound, unanswered questions. Then, let us turn our attention to one of our most intimate senses, smell or olfaction. For decades, the lock-and-key model has dominated our understanding. A molecule's shape fits into a specific receptor, triggering a signal. Simple enough, it seems. But this model struggles to explain certain perplexing observations. Why do some molecules with very different shapes smell similar, while others with nearly identical shapes smell distinctly different? And why do some heavy isotopes, which are chemically identical but vibrate at different frequencies, have different odors? Enter the quantum smell hypothesis, proposed by biophysicist Luca Turin. It suggests that our sense of smell might not just be about shape, but also about the vibrational frequencies of molecules. When an odorant molecule binds to a receptor, it doesn't just fit. It might allow an electron to tunnel across a small energy gap within the receptor. If the odorant molecule has the right vibrational frequency, it can absorb energy from this tunneling electron, much like a guitar string resonating with a specific note. This inelastic electron tunneling would then trigger the neural signal. It's as if molecules are not just shapes, but tiny musicians, each singing a different tune, and our receptors are finely tuned quantum detectors listening for specific melodies. This hypothesis, though still debated, opens a fascinating window into how our most basic senses might be rooted in the quantum realm, transforming the very act of smelling into a quantum event. The fundamental engines of life, enzymes, also present a compelling case for quantum involvement. These biological catalysts accelerate biochemical reactions by factors of millions or even billions, enabling life to proceed at a viable pace. While classical models explain much of their efficiency through induced fit and transition state stabilization, some observations hint at something more. In many enzymatic reactions, a proton or a hydride ion must be transferred. These particles are incredibly light, and at the molecular scale, they exhibit wave-like properties. It is increasingly believed that quantum tunneling, where a particle passes through an energy barrier rather than over it, seemingly defying classical physics, plays a significant role in many enzyme-catalyzed reactions. Instead of waiting for enough thermal energy to surmount a barrier, the proton simply tunnels through it, significantly speeding up the reaction. Enzymes, then, are not just molecular matchmakers, they are quantum engineers, precisely positioning reactants to facilitate this ghostly passage, leveraging the uncertainty of the quantum world to ensure the certainty of life's processes. Perhaps one of the most striking examples, truly an invisible marvel, is magnetoreception, the ability of certain animals, most famously migratory birds, to sense the Earth's magnetic field for navigation. How do they achieve this feat? Discerning subtle changes in a magnetic field that is orders of magnitude weaker than a refrigerator magnet? Current theories point to a stunning quantum mechanism, the radical pair mechanism. Within specialized cells in the bird's retinas, light creates pairs of unstable molecules called radical pairs, each with an unpaired electron. The spins of these electrons are quantum mechanically entangled and highly sensitive to external magnetic fields. The orientation of the Earth's magnetic field can subtly influence these electron spins, which in turn affects the rate at which the radical pairs react. This difference in reaction rates is then translated into a biochemical signal, allowing the bird to quote 42 the magnetic field as patterns of light or dark. It is an invisible compass, etched not in metal, but in the very quantum mechanics of their being, allowing them to navigate vast distances with astonishing accuracy, guided by forces unseen and unfelt by us. These examples, photosynthesis, olfaction, enzyme catalysis, magnetoreception, are not just isolated curiosities. They are profound invitations to reconsider the very nature of life. If evolution has, against all conventional wisdom, found ways to harness quantum coherence, tunneling, and entanglement in the warm, noisy environment of a cell, what else might it be doing? What other fundamental biological processes currently explained by classical models might have a subtle yet crucial quantum underpinning? The implications are vast and humbling. It suggests that life, from its very origins, may have been intimately intertwined with the deepest laws of the universe, not just as a passive recipient of physical forces, but as an active participant, a clever orchestrator of quantum phenomena. It challenges us to look beyond the macroscopic and even the molecular, to truly appreciate the subatomic dance that might be shaping our very existence. How did these sophisticated quantum mechanisms evolve? Were they present in the earliest forms of life, or did they emerge later as refinements? And if life can harness these phenomena, could we, through deeper understanding, learn to manipulate them for novel technologies, for medicine, or even to unlock new forms of perception? As we drift through this dreamy academy, exploring the edges of our knowledge, the quantum realm reminds us that the deeper we delve into life's mysteries, the more profound and interconnected they become. The seemingly solid ground of biological understanding begins to shimmer, revealing a hidden dimension where probabilities guide destinies and particles communicate instantaneously across space. The journey into the quantum whisper is just beginning, and it promises to reshape our understanding of what it truly means to be alive, revealing that the universe's most subtle forces might be life's most ingenious architects. The question is no longer if quantum mechanics plays a role in biology, but how deeply and how pervasively these unseen forces sculpt the vibrant living world we inhabit. Having journeyed through the intricate dialogues of biological communication, from the silent whispers within a cell to the vibrant symphonies exchanged across species, we now lift our gaze to the grandest stage of all, the ceaseless, interwoven drama of ecology and evolution. This is where life's individual stories converge, where the chemistry we explored, the cells we marveled at, and the minds we pondered, all find their place within a magnificent, ever-shifting narrative that spans eons and continents. We invite you, dear companions, to step with us into this vast, pulsating web, to observe the forces that sculpt existence and the profound mysteries that still shroud its ultimate design. Consider, if you will, the very concept of an ecosystem, not merely a collection of organisms, but a living, breathing entity, a complex symphony where every note, every instrument is inextricably linked. From the microscopic bacteria that cycle nutrients in the soil, to the towering redwood forests that reach for the sky, from the deepest abyssal plains to the highest mountain peaks, life is woven into an immense, intricate tapestry. We speak of food webs, of trophic levels, of nutrient cycling, yet these terms, precise as they are, barely begin to capture the profound interdependence at play. What invisible threads connect the flight of a butterfly in one hemisphere to the rainfall in another, or the health of a coral reef to the distant human economy? The butterfly effect is not merely a metaphor in biology. It is a palpable reality, a testament to the fragile yet resilient interconnectedness that defines our living world. How, we wonder, did such a meticulously balanced, self-regulating planetary system emerge, and what are the true limits of its resilience in the face of unprecedented change? Within these vast ecosystems, we witness the astonishing ballet of coevolution, a relentless, exquisite dance between species that has sculpted forms and behaviors across geological time. Think of the predator and its prey, locked in an eternal arms race, each driving the other to ever greater feats of speed, camouflage, or cunning. The cheetah's explosive sprint is a testament to the gazelle's agility, the orchid's intricate mimicry, a mirror of the bee's discerning eye. This dynamic tension, this reciprocal selective pressure, shapes not just physical attributes but entire life histories, creating a breathtaking diversity of adaptations. And then there are the profound, often hidden, mutualisms, the fungi that embrace tree roots in mycorrhizal networks, exchanging vital nutrients, the gut microbes within us, shaping our very physiology. How did these deeply intertwined relationships, where the survival of one species becomes contingent upon another, first arise? Was it a slow, incremental nudging, or were there critical junctures, pivotal moments when two disparate lineages decided, as it were, to cast their lot together, forever altering their evolutionary paths? The precise mechanisms and triggers for these co-evolutionary leaps remain an ongoing inquiry, a testament to the subtle negotiations occurring constantly within the fabric of life. Our journey through evolution often begins with Charles Darwin and the elegant simplicity of natural selection, the survival and reproduction of the fittest. And indeed, this principle remains a cornerstone of our understanding. Yet the tapestry of evolution is woven with far more threads than this singular force. We must also acknowledge the profound, often unpredictable influence of genetic drift, the random fluctuations in gene frequencies that are particularly potent in small populations, sometimes leading to the fixation or loss of traits, not because they confer advantage, but simply by chance. Then there is gene flow, the migration of individuals or genetic material between populations, which can introduce new variations or homogenize differences, constantly stirring the genetic pot. And in the microbial world, a realm often overlooked in macroevolutionary narratives, horizontal gene transfer. The direct exchange of genetic material between unrelated organisms acts as a powerful, rapid evolutionary accelerator, blurring the clean lines of ancestry and presenting a challenge to our traditional phylogenetic trees. How do these diverse forces interact, sometimes synergistically, sometimes antagonistically, to steer the course of life? To what extent is evolution a deterministic march towards adaptation, and to what extent is it a meandering journey, shaped by the whims of contingency and chance? The very pace and pattern of evolution also present profound questions. Is evolution a slow, steady, gradual process, as Darwin initially envisioned, subtly accumulating changes over vast spans of time? Or does it proceed in fits and starts, long periods of stasis punctuated by sudden bursts of rapid change, a concept known as punctuated equilibrium? The fossil record, often incomplete and riddled with gaps, offers glimpses but rarely a definitive answer. We see evidence of both, the slow, incremental alterations in some lineages and the explosive, adaptive radiations that filled ecological niches after mass extinctions, unleashing a kaleidoscope of new forms. We also observe convergent evolution, where distantly related species independently evolve similar solutions to similar environmental pressures. The streamlined bodies of dolphins and sharks, the wings of birds and bats. These remarkable parallels suggest underlying constraints or optimal designs in nature, but they also beg the question, how many paths can life truly take? Are there universal principles, a hidden grammar guiding evolution's hand? Or is each species' trajectory a unique, unrepeatable improvisation? Perhaps one of the most fundamental and surprisingly elusive questions in biology is the very definition of a species and what truly separates one lineage from another. The biological species concept, which defines species as groups of individuals that can interbreed and produce fertile offspring, seems straightforward. Yet, nature rarely conforms to our neat categories. We encounter ring species, where populations living in a loop around a geographical barrier can interbreed with their immediate neighbours, but the populations at the ends of the ring are reproductively isolated from each other. We find instances of hybridization, where distinct species occasionally interbreed, blurring the boundaries. And what of asexual organisms, or those known only from the fossil record? The process of speciation, the birth of new species, remains a dynamic frontier of research. How do populations, once part of a continuous whole, diverge to the point of no return? What are the critical genetic, ecological, or behavioural barriers that solidify this separation? The species problem is not just an academic debate. It delves into the very essence of biodiversity, challenging our attempts to categorize and understand the immense variety of life on Earth. As we stand at this precipice of understanding, observing life's grand evolutionary unfolding, we cannot ignore the profound and accelerating influence of our own species. We have ushered in the Anthropocene, an epoch defined by human impact, where our activities are not merely shaping ecosystems, but actively driving evolutionary change at an unprecedented rate. From antibiotic resistance in bacteria to pesticide resistance in insects, from the altered migration patterns of birds to the rapid adaptation of urban wildlife, we are unwitting architects of accelerated evolution. This raises urgent questions. How will life respond to this rapid, human-induced selection pressure? What does it mean for the future of biodiversity, for the resilience of the ecosystems upon which we ourselves depend? Can we, as conscious agents, learn to guide these forces, to mitigate the negative impacts, and perhaps even to foster a more sustainable coexistence? The grand questions of ecology and evolution are not confined to the past. They are vibrantly alive, unfolding before our very eyes. How much of life's future is predictable, given the fundamental laws of physics and chemistry, combined with the historical contingencies of evolution? Are there limits to adaptation, thresholds beyond which life simply cannot persist? And in the vastness of the cosmos, if life were to arise elsewhere, would it follow similar ecological principles, similar evolutionary trajectories? These are not questions with simple answers, but rather invitations to wonder, to observe with renewed awe the intricate, resilient, and endlessly inventive spirit of life. As our journey through the dreamy academy continues, we find ourselves increasingly humbled by the scale of what remains unknown, yet invigorated by the sheer beauty of the questions themselves. Having explored the deep history and the ongoing dance of ecology and evolution, we are now ready to turn our gaze towards the very edges of our biological understanding, to the profound implications of what we have learned, and to the tantalizing possibilities that lie just beyond our current reach. What awaits us at the frontier of life's ultimate mysteries? Let us prepare to explore the very future of biological inquiry itself,